Okay, so today is the second installment of the Neuroscience and Biology of Belief, and today I will be mostly focusing on things that happen in the body at the physiological level and at the cellular level, but we cannot escape the brain, so it's there. We cannot really do much without our brains. So I will try to center this presentation around Bobby the toddler. If you recall him from the first talk around beliefs, he's the one who is jumping in mud puddles and enjoying himself. And depending on how his caregivers respond to him, he can develop negative beliefs about himself or positive beliefs about the self. So the question is, what is happening, what could be happening in Bobby's body and brain in response to the caregiver's reaction? And the reason why I use could is because it's not fully understand how do we go from reactions in the brain, perceived external stimuli, to actually remembering and the body remembering those stimuli and rehashing those same responses later on. There is some understanding, as you will see, but it's not fully clear. The other thing is why I used could is because the vast majority of research that is happening happens with undesirable stimuli. As you can imagine, to get funding for research, you need to show effect, and to really show effect, it's usually bad effect that people look for. You can study PTSD, you can study the effect of malnutrition or maltreatment or abuse of some kind, but it's really, really hard to get funding for studying the effect of kindness on things. I mean, I found 2 papers, but not much. So most of the research in this area focuses on bad things happening to people. And that will reflect in what I will talk about. So, so the first question will be that I will be addressing is what could be happening in Bobby's body and brain, and how can his body remember and store the cumulative effect of whatever response he had. So the first thing that Bobby can probably feel or sense is mental discomfort, confusion, fear, in case the caregiver had a negative reaction to his jumping in the puddle. In the other case, in the positive reaction case, he could feel excitement, elation. And in a lot of ways, these are physiologically the same reaction. They are heightened states of arousal. And because they are heightened states of arousal, his body will activate the stress response. So if you think about how can this be, try to think about a situation in which you were really, really excited about something happening. You had a party that you really wanted to go to, or you were organizing a wedding, or you were it was your wedding, or your graduation ceremony, and you were really excited, totally high about it. In the end, it still drained your body. You still got toasted by it, and it took you some time to recover. So in a lot of ways, for your body, it is the same as if you encountered an undesirable situation. So in both cases, the body's stress response is activated. The body doesn't know what is happening. It's the brain that decides not a stimulus is good or bad in any given moment. So you might be wondering why I'm saying the stress response is activated in both cases, and that is why. So stress, the definition of stress from the person who coined the term, who was Hans Schalier. He was a physiologist in Chicago some 50 years ago. He defined stress as a non-specific response of the body to any demand made upon it. So if you think about it, in the first case for Bobby, the caregiver's reaction demanded him to kind of maybe tone it down, withdraw, to have a fear response. In the other case, Bobby's body, or the demand on his body was to notice that it was well accepted, that he could go on and he could just keep being himself. So in any case, there was something meaningful happening to him, to which his body reacted. So stress can be thought of as uncertainty, novelty, excitement, and in that regard can be both positive and negative. So that's a very, very important point, that stress can be good or bad. The positive events have the same physiological response as moderate fear or threat. When we need to differentiate really between stress and trauma, as when the stress is repeated, it's chronic, and when it reaches a person's limit to cope with it. And given the work that we do in AD, I think this is an important thing to understand, that trauma is different from stress in that it's over a person's limit. What is a person's limit is highly individualized. So something that will be traumatic for some people will not be traumatic to other people. So in that regard, when we talk about negative life events that can happen to people, the literature treats maltreatment, neglect, emotional abandonment or physical abandonment, parental loss, extreme poverty, all still in the category of stress. Trauma, on the other hand, is listed always as physical or sexual abuse, medical trauma, accidents, so really, really horrendous, terrible things that fall into two categories. They threaten injury, death, or the physical integrity of a person, or causes horror, terror, or the feeling of helplessness in a person. So these are two psychologically and biologically different mechanisms. And I think it's very important to understand that the literature and all the research actually treats these differently, and it's understood now that they activate different biological pathways. So what I will talk about today is mostly stress. Trauma is, we'll leave that for another day, maybe. So the most vulnerable developmental periods to stress are the pre and perinatal periods, early childhood, up to about 6 to 7 years old. And if you recall, that's the time by which our beliefs are being formed. So, and that's when our brain develops at the most. That's when we learn more about the world, and that's when we are still most connected to our real selves. And that is when the brain is being morphed, literally, physically, into what it will become. And then adolescence, when there is another spurt of neurogenesis and synaptic pruning that shapes the adult brain. So stress during these periods can have the most profound impact. So the next question, how is it decided in a person that whether or not a stimulus is positive or negative, whether or not something outside of them happening is positive or negative stress? I usually keep saying that stress is an inside job. You decide, your body, your brain decides how will you perceive a stimulus, whether or not it's going to be stressful or not so. And here we go back to our friends from the last presentation. It's, of course, the default mode network, which is responsible for the conceptual I, the mental understanding of who a person is. Then the dorsal attention network and the central executive that are responsible for rational thinking. So sort of the default mode network and the rational thinking part duke it out who is going to dominate in any given moment, with the help of the salience network, which conveys the bodily sensations and the felt sense of me to these other two networks. So if you recall our seesaw, the slope of this line determines who is, what thoughts a person will have about something, what he or she will be able to cope with in any given moment. And also the interaction of this network of these three networks decides what will come to conscious awareness, what can be dealt with consciously, and what will be just simmering underneath. So back to stress, when the brain decides that there is a perceived or real threat or arousal or something meaningful, the salience, the default mode network activates the amygdala, which is part of the limbic system, which then activates the hypothalamus, which is a very tiny part of the brain right in the middle of the brain, which then activates the sympathetic nervous system. So that's what it looks like. So the hypothalamus, through connection of two neurons, activates the adrenal gland, which then releases adrenaline and noradrenaline and activates the cells in the adrenal gland, which then again release adrenaline and noradrenaline to the bloodstream, and that initiates the flight or flight response. So this is called the sympathico-adrenal medullary axis. Now notice here something that I will come to later, and you will see the importance of it. The nerve cells that come from the brain release adrenaline and noradrenaline onto the cells of the adrenal gland. But the adrenal gland also release adrenaline and noradrenaline into the bloodstream. So these are the very same molecules that they work with. Okay, so that will become relevant later. The other part of the stress response is what's happening inside the brain, also from the hypothalamus. The hypothalamus releases what's called the corticotropin releasing hormone. These are all very complicated, lots of acronyms here. So the corticotropin releasing hormone activates the pituitary, which is this tiny pea-sized gland at the base of the brain, which releases adrenocorticotropic hormone, more acronyms, that reaches through the bloodstream, the adrenal gland. Then the adrenal gland releases, releases cortisol, which then goes back to the brain and shuts down the stress response. So, and that is called the hypothalamic pituitary axis, or HPH, which is usually the main character in the stress response. At least most people consider it. So again, notice here that we are talking about chemicals traveling in the bloodstream from one place to another. It's not direct neuronal connections. So that's a very important message for today, that both in the body and the brain, most of the information is carried by chemicals, and that the brain and body can use the same chemicals. So what is happening in the body when adrenaline is released? Well, one thing is the heart rate increases, the rate of breathing increases, blood flows into the muscles. On the other hand, blood flows out of the skin and the digestive system is shut down pretty much. What does cortisol do? Cortisol increases the blood sugar level, so there would be energy for everything else that adrenaline was doing. Cortisol also shuts down the immune system, because when you are in a stress response, your immune system is kind of secondary. And cortisol, through a negative feedback loop, feeds back into, stops the whole stress response and activates the parasympathetic response. And that whole thing is supposed to bring back the body into a homeostasis. And that's probably how you would learn it in school way back when. It's being recently understood that it's not really homeostasis that we talk about, but rather an allostatic response. And allostasis is remaining stable by being variable. It's a dynamic situation. In homeostasis, as it says, it's stasis. It would be a situation in the body where everything is in the right place, and when it's knocked out of balance, then the goal is to put everything back where it was. And that's not how it works. Things are kept in balance, but by moving parts. And I think that's a very, very important new understanding that became obvious in the last 20 years or so. And at the core of allostasis is a homeodynamic response, which is a dynamic form of homeostasis that involves moving parts, that is based on the interconnectedness of body components, different physiological systems, while the body maintains an overall equilibrium. So to have an idea what that looks like, this image I found really useful. So you have the nervous system, where the red arrow points, the nervous system, the cardiovascular system, the immune system, and everything that has to do with metabolism, the digestive system, and cell level mechanisms. They all talk to each other and they all interrelated. So the arrows all indicate interconnectedness. So this thing needs to be kept in balance all the time. So it's not static. These systems move relative to each other. They talk to each other. So it's a very, very dynamic system. So the homodynamic balance can be permanently changed after repeated and chronic activation of the stress response. Obviously, when such a dynamic system shifts, it very seldom goes back to where it was. It goes to its new normal. And very often people can build up over time homodynamic balances that is not necessarily very good for them, but because of the repeated stress response, that's their new baseline. That's where they start functioning from, and they may not know because it doesn't feel like they are in stress. So the direction in which the homodynamic balance shifts depends on genetic predisposition and people's history of life events. So it's highly individual, and what happened before has a huge impact on what is going to happen in the future. So back to Bobby. So by now we understand that Bobby's body activates the stress response, and he has bodily sensations. As we said, his heart rate changed, his breathing changed, his muscles have a different feel to them. He may feel mental tension. His bodily systems were shifted out of balance for a while. And that means that he is experiencing emotions. And here you might have another one of these, what she's talking about. Emotions, all of that. And indeed, emotions are defined officially as a combination of bodily sensations that are associated with a personally significant event or stimulus. So emotions are not a mental thing. Emotions are a physical thing. And emotions are physiological experiences and states of awareness of the body that give you information about the world. And of course, it's not just the body. There are brain regions involved in processing and storing information relative to these emotions. And of course, it's the good friend. The limbic system, there are some cortical areas involved, and there's the insula, which is part of the salience network, which directly connects to the default mode network. So everything here that I listed in the brain, if you remember from the previous presentation, it is mostly the cauldron of activity that is happening outside of conscious awareness. All these cortical and subcortical systems take part in that, and we have no idea about it, consciously anyway. So emotions can be thought of as a dynamic configuration of brain networks in combination with the body. And these dynamic configurations are created by what is going on in the brain at the moment, what interoceptive information is received from the body, and how the body and brain chat about it. What is the information that being passed from one to the other, how they interact. So these configurations are stored in memory and are reactivated and can be reactivated by the same or similar stimuli or the context that created them. So if you think about when people say my buttons were pushed, it's like these are like programs that we carry. We can think of them as beliefs, right? So these dynamic configurations in the body can be thought of as the biological substrate for beliefs. I'm not saying it's for sure, but that's, for me, this is the best understanding I can come up with. And then, of course, rightfully, you ask, so what are feelings? So feelings are when emotional states come to conscious awareness. So English tends to be rather imprecise about the use of the words emotions and feelings. Not quite every language is that off. But I think it's, given the work that we do, it's actually an important concept to differentiate, two important concepts to differentiate. So feelings are the interpretation of the data. So if you think of emotions are the raw data that the body sends to the brain, then feelings are the interpretation of those data in the context of the stories that we tell about ourselves. Okay. So, and to what extent is it correct that emotions are really in the body? If you think of any body work or somatic work or a massage that you ever had, very often it is accompanied by emotional release. So the body does, and you sometimes you may even not know what a feeling that was, or you cannot even put a finger on what you felt. You just know that something changed your body, and that means that your emotions shifted. So it is an interesting thing that I have to wrap my head around, the difference between emotions and feelings. So now the next question is, how does the body record and remember the impact of a stressful or meaningful event beyond the new balance that can form as a result of the interaction of the various body systems? So the body records and remembers things through emotion. And how does the body and brain communicate about these emotions? We saw that they do so through chemical messengers. So I will not go into much detail about chemical messengers because that would be brutal and cruel to everybody. But there are two major types of messengers, neurotransmitters and hormones. And this is the traditional classical division. And this has to do with mainly where the compound was discovered. So there are compounds that are called neurotransmitters historically because they were discovered in the brain, and hormones historically because they were discovered in the body. But what has become clear is that most chemical messengers are both neurotransmitters and hormones. And I did a game with this for myself, and I checked the most obscure neurotransmitters that I could remember and checked if they are found in the immune system or in the cardiovascular system, and indeed they are found there. So this is the rule rather than the exception. There might be exceptions, but probably not many. And this means that the brain and the body speak the same language, and that information is carried back and forth via chemicals. The brain and the body primarily speak with each other in the form of chemical messengers. So if chemical messengers are everywhere, and there are so many of them, where is precision in this communication? That's a very valid question. And that hinges on receptor molecules that are on the surface and inside the cells. So you are all familiar with the cell, there's a membrane on the outside. There are many, many components on the inside called organelles. And the center is a nucleus, which contains the DNA. Now, outside on the membrane, there are these cell surface receptors or transmembrane receptors, which come in many different shapes and sizes, but there are very definite categories. So the first one, the red one, is a 7 membrane spanning with 7 different subunits. I didn't name them because this is just showing its decoration mostly. It's showing the many varieties. So the role of the combination of receptors on the cell surface is to accurately read and interpret signals from the environment and keep track of activation, of its own activation history. I found this rather interesting, that apparently receptors can remember how many times they were activated in the past, and that influences their sensitivity and responsiveness as a result. So if you think back to repeated or chronic stress response, if a receptor on a cell is activated over and over time, it will start responding differently. Receptors tell the cell what to do. And the cell can be in one of two states. It can grow, heal, restore itself. It's moving forward type of activity, or it can be stimulated, activated in stress, where it's more like in a protective mode, where it's surviving, but not really much of growth is happening, or moving forward, or improvement, let's put it that way. So how do receptors work? So when you work with receptors, you have two parts. You have the receptor and you have the ligands. And originally the thought was that it's the best model to describe receptors and ligands and their mechanism of working is the lock and key model. So you would have some ligands that fit into the receptor like a key into a lock. So here A fits into the lock and B does not. So A can activate the receptor or interact with it in a meaningful way, and B cannot. So the interesting thing is that it appears that both ligands and receptors are capable of changing their conformations. So these are all quite complex chemicals, so don't think of just one simple, not just a few atoms. These can be quite big molecules, particularly the receptors, very complex. So they are able to change their conformation really, really fast. And it is apparently really hard to tell whether or not they are in one state or another. They do this mostly through sugar molecules or lipid molecules. And I read it from a very reputable source that in some way they can behave both like a wave and a particle, that they can change their configuration so quickly that you cannot tell which form they are in at any given moment, which gave me the thought that this is probably where their impact from energy, be it electromagnetic fields or quantum fields, can come in. I'm not saying it is, I'm just saying there's a possibility. I found this a very interesting fact. So receptors are very, very numerous. They come in several different types. The easiest to see how it works and easiest to understand are the ligand-gated ion channels. So what happens is that the ligand, a signaling molecule, which we can call the primary messenger system, and these are the primary messenger system are all the chemicals that are floating around in the bloodstream and in between cells, and they are the ones who carry the information. So the ligand binds to the receptor and it opens or closes, and depending on that, it lets ions flow through. And these ions are usually potassium, calcium, sodium. And ion channels are very prominent in the nervous system, and they act in a millisecond range. They are very fast. They are the means through which neurons talk to each other very quickly. Then the second type of receptor are the G protein coupled receptor, which can do many, many things in the cell. In this case, it shows that indirectly they interact with an ion channel, but they activate second messenger systems inside a cell, which are very complex combination of more proteins that talk to each other. And the range of response time for G protein coupled receptors is in seconds. And these are found in all cell types. Then there are enzyme-coupled receptors. Enzymes are like catalysts. They make certain chemical reactions in the cell happen. They are also in all cell types, and the response time is usually in minutes or hours. And finally, there are receptors that are found in the nucleus. These are called intracellular receptors. And for example, cortisol, part of our stress response, is such a ligand. It goes through the membrane unimpeded. It's a lipid, right? It goes through the membrane, which is made out of fat molecules, goes straight into the nucleus and affects gene expression. The last three, yes, and it takes hours for the response because it affects gene expression. That's a slow process, relatively speaking. So the last three receptor types are usually the ones that play a role in development and plasticity of cells. So the G protein just want to show you roughly what the G protein coupled receptors do. So you need to know there are more than 1,000 types of them. They are quite conserved in terms of their structure from organism to organism. So evolutionary, they are quite conserved. And what they do, so you see here these colorful, these columns, those are the subunits of the G protein coupled receptor, and they activate a second messenger system cascade. This is a relatively, I would say, not very complicated one. We don't need to know what really happens here, but I wanted you to see the complexity of the biochemistry that is happening inside a cell once such a receptor is activated. The bottom line is that more often than not, the second messenger system cascade ends up in the nucleus, influencing gene expression. The second one I want you to see are the nuclear receptors. There are much fewer types of these, only about 50. They cater to steroid hormones such as cortisol, and all the sex hormones fall into that category as well. And what these guys do is the hormone enters the cytoplasm, binds to the receptor, goes into the nucleus, and directly acts as a transcription factor for transcribing genes, meaning making a messenger RNA from the DNA. So just to sum it up, receptors are specialized membrane-bound proteins. They receive external stimuli, which are called ligands. They undergo structural changes after binding a ligand. Binding the ligand triggers various events inside the cell. These serve as, essentially receptors serve as signal transducers across the membrane. So whatever information the cell received on the outside is conveyed to the inside by the receptor, and it's the receptors that decide how the cell is going to respond. And very often, the same ligand can have an opposite reaction on a cell depending on the receptor type. And I think this is, before we go there, this is the guessing game for the day. How many receptors do you think are on the surface of a cell? If you want to type it in, it's truly a guessing game. 3.1, yeah. Yes, I think Ron is the closest, way above, probably even. So I heard numbers floating around, but it's apparently in the millions. And because we have 100 different types, and some can be in the 100,000s of each, we can easily have millions of receptors on the surface of the cell. And you can think about them as floating islands in the membrane. So they move around. It's not like they are static. So they can interact with each other. They can congregate together. It's a very dynamic system on the surface of a cell, how it receives information from the outside. And so we can move on to the nucleus now. What is happening in the nucleus on the genetic level is determined by genes, which is the sign of genetic predisposition, or availability of genes, which can depend on developmental stage, or the environment, which is what we call epigenetics. And epigenetics is a relatively new idea. New as in about 30 years old, probably, since it's understood how it's really the environment that determines how organism develops and how an organism responds rather than its genetics. Genetics plays a role, but it's not in the driving seat. Genetics provides the raw material, the blueprint, but it's really signals from the outside that run the show. So to understand how what happens at the DNA level, we need to start with the three-dimensional organization of genes. So this image that you are all familiar with is that of a chromosome, and it's a tightly packed DNA, which is inactive, and you see the chromosomes like this only during cell division. And we have 23 pairs of them. The way they really look like in an active cell that is not dividing is they look like noodles, really, and they're called the chromatin. And the further structure, so you can see the blue thing, which is the DNA. So the chromatin is made up of DNA and histone proteins. So histone proteins are specific proteins that are bundled together. There are 8 of them, and the DNA is wrapped around it. And these wraparound things are called the nucleosomes. So the way it works is like beads. So you have the DNA is running, it is wrapped around the histones, that's a nucleosome. Then you have, again, a naked piece of DNA, and then you have another nucleosome. So it's like beads connected to each other on a thread. That's how to imagine the nucleosomes. And that's called chromatin packaging. So that's one level on which the cell, the outside signals determine what's happening inside the cell. The second epigenetic modification is chemical modification. Most commonly, that's methylation, which is just plain adding a methyl group onto the DNA or onto the histones. So I don't know if you can see it, but histones have little thingies coming out of them. They are called the histone tails, and they love to accept smaller molecule of some charge, and methyl groups are the most common. So chemical modification can have a huge impact on whether or not the DNA is available to make messenger RNA and therefore proteins from it. As it turns out, the more methylated the DNA is, the more able it is to be active. So when we are born and start to develop in a blastocyst and zygote, and our cells are still pluripotent, our DNA is highly methylated. As we age, we keep losing methylation. And the same way, any negative impact on the cells is removing methylation. So the more methylation, the more available of genes. And then the next level of epigenetic modification can come with gene transcription. So transcription is the term that is used to describe copying of messenger RNA from DNA. And so to look at it, the orange thing would be the DNA, which has a gene on it, gene B, which is green. And every gene has regions before it that are used to regulate its activity. And one is the promoter region, right up next to the gene, and there are distant control elements, which can be of many, many different kinds. So when a gene is being copied into messenger RNA, this is what happens. So there is a bending protein that folds the DNA so that the regulatory regions, the distal control elements can come close to the promoter, and there are all sorts of proteins packed together between the two strands of the DNA. Well, they are not the two strands of the DNA, they are the two regions of the DNA. And then this is called the general, so what's in here in the ovals are general transcription factors, co-activators. There's a molecule that's called the mediator. So imagine that in between these two parts of the DNA, there are at least 50 different molecules packed in that influence how a gene is being copied. And many of those are subject to modification, also often by methylation or by some structural change. So it's very common. I'm not going to go into any details. But for example, there's a molecule called the mediator, which has 38 subunits, I believe, and several of those subunits can be altered in a way that influences how it then influences gene expression. So it's a very, very complex cascade of events that can happen with gene transcription. Yes, and this is called the transcription initiation complex. And when that's activated and the gene is ready to be transcribed, then the two DNA strands separate and mRNA is being copied. And so the constellation of proteins in this transcription machinery and their modifications are the epigenetic level of control. Yet another one. Then there are messenger RNA modifications that can happen. And one of these modifications is simply removal, and that is degradation by non-coding RNA. So all of you probably heard of the fact that approximately 99% of our DNA does not code for proteins, and that's called junk DNA. Junk DNA can do many, many things, one of which is that it codes for these non-coding RNAs that come in a long, micro, and tiny form. They are just indicating different length. And all they do is they bind to the freshly made messenger RNA and break it up. And that's another level of control. And it's again very complex, what the long ones do, what the micro does, what the tinies do. So it's quite a process to figure out how each gene or how each messenger RNA can be further modified. And then, of course, for messenger RNA, also there is chemical modifications, similar to DNA. So the next level, the next thing that happens in a cell, you take the messenger RNA and make proteins out of it. And the question is there epigenetic influence there? And the answer is yes. Once the proteins are made, they can also be methylated. And as I just mentioned with the mediator protein, with the histone protein, then through that methylation, they impact what happens in the cell. In addition, there can be epigenetic modification of mitochondrial DNA, which you all know is influencing how much energy a cell can use and how it uses energy. And then finally, there is telomere length, which most of you have probably heard of, that at the end of the chromosomes, there are these regions called telomeres, the length of which is correlated with the health of a cell or health of an individual. And the longer the telomeres, the better. So illness, stress cuts down on the length of the telomeres. That also counts as an epigenetic modification. So epigenetic changes produce heritable changes. It's a sort of an intracellular memory. They influence responses to later stimuli. As you can see, changes to the DNA, changes to proteins. The cells don't lose that very quickly. They can go on from generations to generations within a cell, and some of them is heritable from one person to the next. So you can be influenced by stress that happened to your mother, any epigenetic modification that happened to the egg from which you formed it will show up in you. Also, they are not a static thing. This is just like the rest of the responses I talked about in the body. Epigenetic modifications are dynamic. And they are found in every cell, but most profoundly in the nervous system, especially in the hippocampus and amygdala, which have to do with memory, and both of which primarily memory and meaningful events that we consider meaningful events, processing of meaningful events. So, and I won't go into how things are different in the nervous system, because if we talk about it, it'll be under memory, because that's where it's most relevant. So the final question really is, are these epigenetic modifications reversible? And the answer is yes. Funnily, there are molecules called readers, writers, and erasers of epigenetic information. These are their official names. So obviously, they, as the name suggests, they add, decode, or remove these modifications. And what is beginning to become clear is that that it is possible to undo the damage that is being done by stress on the cellular level by kindness, by love, by thinking positively, by not dwelling on things. So whatever information I could find about changes to epigenetic modifications suggested that yes, changes in behavior, changes in thinking can reverse prior adverse events. And basically the take-home message for me from this was that it really does matter what environment we embed ourselves in, what information we interact with, what information we allow ourselves to take in, and how we react to that. So being mindful of what media we consume, who we hang out with, can all have a huge impact on how our cells are and how our body is feeling, and what beliefs we keep and how we change them. And that's it for today. Thank you all. Any questions?
That was great. Thank you. So the junk DNA, I guess that's the epigenetics. That's from your ancestors and from early childhood memories.
Epigenetics can be anything from your ancestors to your childhood to yesterday's lunch.
So it's not junk DNA.
The junk DNA is the part of DNA that codes for some of these messenger RNAs. So think of DNA, we can go back to, we don't have to. So think of DNA as a long string, right? That is then wrapped up into the chromatin. And at some point, it was believed that all of the DNA will code for a protein. Before the human genome was sequenced, it was believed that we will have hundreds of thousands of genes that each code for a protein. And then when the human genome project was finished, people realized that, oopsie, 99% of the DNA does not code for any protein, right? So that's why they called it junk. And they didn't know what that was, what it was doing. So why is it that only 1% of our DNA codes for anything that we can recognize, can wrap our heads around? So it's still not fully understood what it does. In spiritual circles, there are theories about it, right? If you look into ion work and stuff, but also these, and the junk DNA is always there. It's part of your chromosome. You inherit that with everything. It goes from cell to cell. You got it from your father, you got it from your mother. When it impacts the epigenetics or your current functioning is when it codes for these when these non-coding sequences are actually copied into RNA, and that RNA, that non-coding RNA, interacts with a messenger RNA, and messenger RNA always codes for proteins, right? That's what we understand as, that's the meaning of a gene, that it's information that allows a protein to be made via a messenger RNA. So you have the messenger RNA here, and then these non-coding bits of RNA come in and mess with it, literally. And again, I'm not thinking that all of that 99% of the junk DNA codes for these regulatory little messenger, the regulatory epigenetic RNA pieces. So who knows, but that's what it's coming from. So think of it as junk DNA is always there. It's part of your chromosome. It's doing something, but it's not coding for protein.
Okay, thank you.
Did it make any sense?
Oh, yes, yes. Mm-hmm.
Okay.
Melinda, I'm wondering, of all the information that you presented, what do you feel are the most important takeaways relevant to the work that we're doing with beliefs?
For the work that we are doing, that there is a difference between emotions and feelings. For me, there is a difference between stress and trauma. I think that's a very important thing. There is I'm realizing there is certain level of trauma that I would not touch, or would be very, very careful about. So there are certain things that people carry that might not be amenable for the work that we do in AD, because we don't know what can of worms we open, and they may have psychological patterns that we cannot work with effectively, or should not touch. So I think it's above our pay grade, honestly. So somebody with severe sexual abuse, I'm not sure I would take on, for example. That I would just be very careful. The other thing, so beyond these conceptual distinction, I think it's important to understand that the brain and body really does use the same language. So what we think and what happens in our brains impacts our body directly, and what happens in our body directly impacts our brain. And this is one reason why movement is so important and so effective, is that what we do with our body will have an impact, and what is done to our body in terms of, say, a massage or a body work or something will have a huge impact on our brains. So we cannot really, draw a line here. It's like, oh, I'm not working on my body, on my brain, right? No, the two have to be addressed together. They are in cahoots together, and that's how we need to treat them.
So you can be epigenetically affected by stress that occurred to your mother while she was pregnant with you.
Yep.
And since it is reversible, awakening dynamics work should be effective in helping a lot, which is why our stress or our trauma protocol works.
Yes.
Right.
Yes. And this is why we find you have an ancestral something, right?
Yeah.
And it's quite common. And this is one way in which we have that. Yeah.
And anything, good grief. I mean, people that don't want to be pregnant, teenagers, I mean, good grief, the babies they are having, they're just born with all kinds of stress.
Yes, absolutely.
That we know nothing about as we grow into adulthood.
Yes. Yes.
But it's still there.
Yeah. And if you think about your grandmother, in whose body the egg was formed that your mother became, so you are actually carrying even from your grandmother.
Right.
Right?
Or beyond, which is like
beyond, exactly.
the ancestral effects.
Yes. Yeah, so and this is why history matters, like what the person's history and what the cell's history was, and clearly what the receptor's history was, which I found really curious. It all adds up.
Awakening dynamics work it's really working on all of our cells.
Yes. Yeah. Yeah.
Quite amazing.
Yes, I thought so. How the different layers all connect, and
yeah, layer after layer after layer. I mean,
yeah,
that's why it takes more than one session.
yeah, and this is why many things can come up over and over sometimes, right? Because you address them on a
right. You take away one layer of problem, but then there are others still waiting to be dealt with.
Exactly. Yeah.
I see.
Melinda, if I could ask, when another reference to this is previous life contracts, are we talking about the same thing? Is this the same thing?
I think previous life contracts would be more on the energetic level. I'm sure they show up somewhere in the biological form, but I cannot tell you how. If you just think about it, that in reality, we are a projection. So we are an energetic being. And what I explained was mostly biology, the way science understands it today. I honestly cannot tell you how to go from one to the other. I have no doubt that energetically we carry these past life contracts or soul contracts or any other energetically existing contracts. But how it translates into biology that we understand today, unfortunately, I can't tell you. The closest I could get with anything woo in this was that the receptors are actually both a particle and a wave form. So that was the closest that came to anything quantum field level type thing. And I'm sure there's a lot more. We just don't understand it. We can't measure it, can't detect it at the moment.
There was a fellow name, if you know him, his name is Gary Null. He's been around forever, and he's on either his 5th or 6th clinical study of adding years onto your life, and they use the DNA methylation test to do it, and it takes a couple months to do that testing, but you can actually tell your biological age through this testing, and in just over I think maybe a month or 2 months of work, people have added 5-6 years to their life, and that's just 2 months of de-stressing yourself, exercising, diet, and repurposing your life simply by doing that. He's added. Yep. Have you heard of him?
Yeah. Yes, I did. Yes, methylation is huge, and it's fascinating that it's on every level, and it's always methylation. Well, I mean, there are other chemical modifications, but it's like 80% is methylation. Yeah, I heard of it. Yes, and I do find it fascinating.
It is. I do too.
And I also find it fascinating that I would say 30 years ago this was new. So as recently as that. So yes. Okay. Any more questions? No? Okay. Well, thank you all for coming, and it's time. And until next time.
Thank you. Great presentation.
Thank you. That was awesome.
Thank you. I hope I can teach, say something that is useful and teach you something that is useful.
I can't wait to watch it again. Yep.
Yeah, I need to watch it again too, considering my computer crashed midway.
Okay. So here I am.
Thanks so much.
So I aim to, present you guys with something that is useful, and yeah, that's all. Okay, so see you next time, whenever that will be. I don't know yet.
Okay, thanks again.
Okay. Bye-bye.
Bye-bye.